For joining us today. We're very excited to present MegaSporeBiotic to you. It's quite a unique product in terms of a probiotic, and then we're going to talk about why that's so unique and how it really fits the true definition of what we now understand to be a probiotic. There's a lot of new science surrounding the understanding of the gut microbiota, probiotics, their form and function, and this is really the first product that fits into that new paradigm. So to start, we'll go just do a quick overview on the history of probiotics. It was first discovered in the early 1900s by a Russian scientist named Mechnikov. And really what he looked at is fermented foods. And he realized that fermented foods had bacteria in them that were beneficial. And fermented foods, the bacteria provided different benefits to the human physiology. And he then coined this idea that there are bacteria that are good for you versus bacteria that are all just bad for you. The term probiotic itself was really used in the 1960s, and since then obviously is grown to be pretty well recognized as a term amongst healthcare practitioners, consumers, and the like. For the most part, probiotics claim to help with digestive health, digestive discomfort, and of course some immune support. But we'll show you that with a true probiotic, the usage and the applications are far more extensive because so much more of your physiology is controlled by the gut than we actually even understand at this point. And that understanding is very quickly changing. The current market size in the U.S., it's over 775 million in sales with a 22% annual growth rate, and that's just in the dietary supplement capsule form. They estimate by 2016 that it should be upwards of 1.5 billion in total sales in capsule form, and really this year alone, people are estimating that there's $145 million in new sales in probiotics, and the idea is how can we help doctors and healthcare practitioners capture more of that market versus it going to the large retail outlets like the Walmarts and Walgreens of the world. Food-based probiotics sell for about $30 billion a year, and that's mostly in yogurts and increasingly in kefirs and some probiotic drinks. But is this really justified? Are yogurts really a source of probiotics? And I think you'll see where that answer comes in a few slides. Current market leaders, most probiotic supplements are sold in the retail outlet. Here are some of the current leaders: Culturelle, Align, Philips Colon Health. These are some of the top brands you find in the retail space. And over 90% of leading products utilize Lactobacillus and Bifidobacter species. Those are the most common used probiotic species, and we'll talk about why there's issue with that as well. Now, I mentioned in the beginning that there's a totally new understanding, new paradigm behind the gut microbiota and the understanding of what a probiotic is, what our microbiota does, and how it functions. All of this is coming out of a project called the Human Microbiome Project. It was a project initiated by the NIH a little over 5 years ago. And in fact, some of the data just started coming out in 2012 and some additional in 2013. Why do they use the term microbiome? Well, the microbiome is defined as a totality of microbes, all their genetic elements, and the environmental interactions with their host, which is the human body. And you may ask, well, why did they put together this big project, the Human Microbiome Project? Well, to begin with, the human flora has never really been completely characterized. There's a lot of limitations in microbiology in the decades previous. Microbiology uses growth media and uses swabbing techniques to grow up bacteria to characterize them. The problem with most bacteria that live within the GI is they're obligate anaerobes, meaning they don't survive in an oxygen environment. So when you pull them out of the GI through fecal sampling or whatnot, and you try to grow them up in a petri dish, they don't end up growing. They die. In some cases, they die within seconds of being exposed to outside air. And so it's been very difficult to really characterize what microbes actually exist in the gut and what concentrations and how are they distributed. Now they use a technique called metagenomics. Metagenomics is an advance in RNA and DNA sequencing, which really allows for pinpoint accuracy in terms of characterizing each and every species. It uses a section of the RNA of every bacteria called a 16S ribosomal RNA. It's essentially a fingerprint for every single species of bacteria. So by looking for the 16S ribosomal sequence in a sample set, then you can identify what bacteria is there, and you can even identify what amount of bacteria is there. So this is a very, very accurate way of doing it, and it doesn't matter if the bacteria is alive or dead. Again, as I mentioned, it was launched by the NIH a little over 5 years ago. Currently, there's over 200 researchers involved in this project in 80 different research institutes, so we're getting a lot of work done understanding the role of the microbiota and the form and the function. So here are some facts that we've learned from the Human Microbiome Project, and this really plays well into our understanding of what a probiotic should be. Number one, we are more bacteria than we're actually human. There's actually 10 trillion human cells that make up our entire body, and there's over 100 trillion bacterial cells that inhabit our body. So we're almost 10 times more bacteria than we are human. There are 1,000 different species of commensal organisms in the gastrointestinal tract of each individual, and that is out of 35,000 possible species. So what that tells you is that everybody's GI is essentially a fingerprint. You know, if you've got 1,000 species in your GI out of 35,000 possible, the likelihood of one person to the next having a very similar, if not identical GI is almost non-existent. So everybody's GI is quite a bit different, and it's shaped by how the GI was seated, their body's interaction with the microbiota, their diet, their lifestyle, and many other factors. Bacterial genes outnumber human genes 150 to 1. And that's actually a very, very compelling piece of information, because one of the issues that came out of the Human Genome Project, as you know, that was launched over almost 20 years ago, was that scientists had estimated that we probably had around 130,000 genes in the human DNA in order to conduct all the metabolic functions that a body does. But once they've sequenced the whole human genome, they figured out that we only have 25,000 human genes. That doesn't really account for all the metabolic functions that the human body is capable of. One of the answers is that we are potentially using microbial genes to conduct metabolic function. And now they have shown examples of this. We actually use bacterial DNA in our GI. We absorb it and actually utilize their DNA for our own cells to conduct metabolic functions. So our relationship with the microbial population in our GI is far closer than we ever imagined. As I mentioned, no two individuals have the exact same composition, not even twins. They've done a lot of characterization on identical twins. At the phylum level, it's conserved. If you're familiar with the taxonomy, starting with the kingdom and phylum, going all the way down to the species. So at the phylum level, it's fairly conserved, but when you get down to the species level, it can be quite a bit different. They have confirmed that disease is associated with disruption of gut ecology. Of course, people in the natural health field have always assumed this and always understood this. Hippocrates had actually estimated this back in 500 BC. But now modern science is validating this, that many diseases, things like Crohn's disease, IBS, asthma, even obesity, are all related to disruptions in the gut ecology. Pathogenic organisms are normal inhabitants of the GI. Bacteria like Helicobacter pylori, Staphylococcus, E. coli, we're all very scared of them, you know, always thinking about looking at how to kill these bacteria, but in fact they are normal inhabitants of the GI. They create very, they perform very key functions. H. pylori, for example, factors in your leptin function. Leptin is a big weight maintenance and hunger and satiety, and so H. pylori actually plays a big role in weight gain and weight loss and maintaining a healthy weight. The problem with these pathogenic organisms is that when you have dysbiosis and they're allowed to outgrow the numbers that are safe, that's when you start to get an infectious situation. The other thing they figured out is intestinal microbiota is not homogeneous. So it used to be understood that the digestive system really is kind of a big vat of fermentation. It's just, like a big vessel. It has a lot of bacteria in it, and there's a lot of non-specific fermentation going on. But really what we've seen now is the distribution of organisms and where they're located in the GI is very, very specialized. And that's an important factor when considering probiotic therapy. So if you look on the left section here at figure A, we're looking at a moving vertically from the stomach all the way out to the colon. You see there's very different populations of bacteria moving vertically, and then also different species that inhabit those different tissues, those different areas in the body. If you look at figure B, this is looking at a cross-section of the GI, looking at the intestinal lumen and moving distally from the lumen all the way to the epithelial surface. You see through the mucosal layer, there's very different bacteria, very different microorganisms that inhabit these different tissue layers. And one may not be suited for a particular tissue layer, and so the distribution of microbes through these tissue layers are very, very specific. In figure C, this is a very important figure as well. This looks at where did your microbiota come from. At birth, the body becomes, the infant, the neonate becomes inoculated. And in fact, now they're finding that in utero there is some inoculation going on as well. They're finding mother's gut bacteria in cord blood, in amniotic fluid, in utero. And so it used to be thought that in utero it's completely sterile and the baby's not exposed to any bacteria until it comes out into the world. But that's not true. It seems like there's very specific transport of mother's GI bacteria into the fetus through the cord blood and through the amniotic fluid, which means the fetus is swallowing it. And so that, in fact, prepares the fetus for passage through the birth canal. Through the birth canal, there's a large inoculum that basically inoculates the fetus's GI, skin, and everything else. And then once the baby is born and starts breastfeeding, that's where a large amount of the gut commensal organisms is passed. There's over 400 different species of bacteria found in mother's milk. So mother's milk, and the other thing is a big component of mother's milk is an oligosaccharide that the baby cannot digest. And the only reason for the oligosaccharide to be there is to actually seed the GI. So the body has gone through some very specific designs to make sure that the fetus and the neonate, the fetus and the infant is inoculated with mother's gut bacteria. This brings up an issue that it's so important for pregnant women to optimize their GI and their gut health, because that's what they're passing on to their children. The Human Microbiome Project. The microbiota communicates with and influences human cell behavior. They do this through various signaling molecules, recognition of bacterial epitopes. Those are, constructs on the structure of the bacteria, outer structure of the bacteria, and this is done both by the epithelial cells and mucosal cells in the immune system, and immune cells as well, all within the epithelial and mucosal layers. As I mentioned before, not all species of bacteria are able to or designed to inhabit all tissue types. So looking at probiotic therapy, just throwing in random species of bacteria into the GI doesn't necessarily get you the specific effect that you're looking for in terms of immune activation in the mucosa, the epithelial layer, so on. So having understanding of what the gut microbiota is structured like and functions, begs the question: what kind of probiotics then are required in order to be effective? And we have some idea of that already. First, there are three major features of a probiotic that are what we term required features of a probiotic in order to be classified as a probiotic. For one, they must have a position in the microbiota. Considering there are nearly 100 trillion cells in the GI, And all of them have very specific binding sites, as you see, as you saw from a couple of previous slides. They all inhabit very specific tissues. It is important for a probiotic to be able to colonize well, meaning it has a spot in your GI. If it doesn't have a spot in the GI, it will not be able to colonize because it will basically get kicked out by the 100 trillion other bacteria that are in there. So there has to be a very specific tie to that bacteria that you're taking in as probiotic and whether or not it has a place in that individual's GI. The second rule is it must have sufficient numbers to colonize and cause stimulatory benefit. So in large part, probiotics cause their beneficial effect by stimulatory mechanisms, meaning they stimulate the immune system. When they're there in large enough numbers, they can create enough of an immune stimulation to give you the positive effects. They also require enough numbers in order to produce some of the molecular compounds that they produce, digest the food the way they're supposed to digest to help the host, and also to have enough numbers to ward off infectious or opportunistic infections. So you have to be able to supplement with enough of that particular species where they can actually get in and perform those functions. Also, supplemented probiotics by evolution should naturally occur in the environment because this is how they gain exposure to the host. In our modern day, in our modern food system and supplement system, we've got wonderful capsules and bottles that we can take on a daily basis. But you have to ask yourself, where did our ancestors get their probiotics? Clearly, they didn't have a Whole Foods or Sprouts with supplements 10,000, 20,000 years ago. Yet these bacteria have evolved to coexist with humans over the last 100,000 years. So where did our ancestors get their probiotics from? And the only answer to that is through the environment. They gained exposure to these beneficial bacteria, these commensal organisms through the environment. So any true probiotic should be found in the environment and should naturally be able to survive through the gastric system in order to colonize in the GI. So here are our current solutions in the dietary supplement industry for probiotic therapy. The product on the left is Supplement Facts of a very popular retail product. I think this product's called Ultimate Flora. They market 50 billion cells as a daily dose, as an effective daily dose. They've got, I believe, 10 strains in here, various strains of Lactobacillus and Bifidobacter. And then on the right-hand side, you've got Supplement Facts, another equally popular supplement product that has right around 15 strains, I think. But they describe a daily dose of 21 million CFU. So what's really interesting about this is both these products exist on the same shelf. And one product is claiming 50 billion is the effective dose. The other product's claiming 21 million is effective dose. They have a lot of similar strains within them. That begs the question already, well, which one's right? How can they both be effective with such different dosage? It's like a company selling a vitamin D product claiming 10,000 IUs is the effective dose. Another company right next to it selling and saying 1 IU is the effective dose. It doesn't make sense. So really, this is not based on any sort of scientific information. The other solutions right now in the dietary supplement category, here's a product on the left, which is again a very popular retail product. They only have one strain in them, very, very specific strain, and they deliver something like 1 million cells. And then of course yogurt on the right, which is some of the biggest area, Activia being one of them. And in fact, Activia actually lost or had to settle a class action lawsuit against some of their probiotic claims. But these are the current solutions available to most consumers in the dietary supplement industry. Here are some of the issues with the current offerings. Number one, this graph shows the number of publications related to the microbiota and the understanding of gut microbes over the last 20 years or so. If you look at the number of publications just in the last 4 or 5 years, it's much higher than what has been done over the last 10 to 15 years. Most of the products on the shelf or in the market today are based on the science in the late 80s and early 90s. But that's just old science. Most of the products now have to evolve to match with what science now understands about how these bacteria function in the microbiota. Other issues with current solutions in dietary supplements. Some strains used in those kind of products that have 15, 20, 30 strains in them, some strains used may not have studies to show any benefit at all. If you're just putting together a formulation with 15 different probiotic strains in it, how do you know if any of them have any efficacy unless each of them have been studied separately? There's rampant mislabeling of probiotic strains, and this has been found in many different publications. There are several documented cases of mislabeled products. An EU panel of experts actually went around and pulled together a number of the most popular probiotic products and characterized the different strains in there to see if they actually match what was claimed on the label. What they found that, number one, products were incorrectly labeled with the wrong strains. Nobody really knew what was in any of these products, and that's not hard to see when you consider how a lot of these manufacturers get their probiotic raw material. You could pick up the phone and call a distributor out of China or somewhere else and order a big drum of various lactobacillus strains. It comes in, a lot of times it comes in pre-mixed, and there's a label that shows that there's 10 different strains in there, but nobody's taking that raw material and sending it to a university lab and characterizing whether each one of those strains actually exists in that big drum of powder. And in fact, if you try to do that, that's like a 6-month university project. It's not a simple, quick assay in order to do that. So nobody's doing that. So nobody really knows what's in these mixtures that are coming. And that's being shown in some of these kind of studies. Products will often contain markedly reduced numbers and claim than what was claimed on the label, which is not surprising to us, and we'll show you why. And often they found the presence of strains that are not at all labeled. In some cases, these are strains that are pathogenic. So really, in many of these products, you don't really know what's in them. There are at least 3 other publications that support these findings, and if you really start digging into it, you really start finding that there's a lot of different publications that demonstrate the same issue with current probiotic offerings. Here's a really good study we like to illustrate. The Food Standard Agency in the UK, in collaboration with Reading University, performed this test. Because, as I mentioned before, most of the strains that exist within the GI are obligate anaerobes. They don't do well out in the outside environment, and this applies to Lactobacillus and Bifidobacter species. Obligate anaerobes that are gut commensal organisms, they're transferred from mother to child in utero, through the birth canal, and through breastfeeding. They never leave the body and re-enter. And so they're not really, from a scientific standpoint, estimated to be able to survive being grown in a big factory and then encapsulated and then taken in orally. So this is one of the studies that looked at some of the most popular strains of Lactobacillus bifidobacter species to see if they actually do survive the gastric passage, because they're not designed to do that. They took 35 strains of commercial products, primarily, again, Lactobacillus and Bifidobacter species, and they evaluated the survivability through the GI. The first step, they put all these strains through a pH of 1, which is not an uncommon pH for the stomach. At a pH of 1, none of the strains survived. Every single one of them was killed off. So if your GI is around 1, 1.5, all these strains would die immediately. At pH 2 to 3, 18 of the 35 showed some survival, but about half their numbers made it through. Then they took those survivors and they put them through upper GI bile acid tolerance testing. Of the 18 that survived at pH 2 to 3, only 6 showed any viability through upper GI itself as well. Then they took the 6 and they put them through lower GI conditions, and of the 6, only 4 strains showed any viability in the lower GI. And remember, the 4 now have less than half of the original cell count that they started with. So at the end of the day, of the 35, only 4 strains made it to the site of colonization, and even the 4 had half the numbers that they actually started out with. Now, to follow up with this study, we've done our own. where we hired Silica Laboratories out of Chicago. It was a world-renowned laboratory, and we asked them to test the survivability of some of the most common sources of probiotics today versus MegaSporeBiotic. And again, remember, MegaSporeBiotic is not enteric-coated, there's no special technology to help it survive the stomach. These are just bacteria that are naturally designed to survive because that's how they've evolved. And so that's this study right here. 4 main products were tested. The first one is a leading retail brand supplement, the Ultimate Flora product with 50 billion cells. If you go into Whole Foods or you go into Sprouts or any of these stores, you see this product, all over the place. It's one of the leading top sellers over there. We also looked at Chobani Greek yogurt. Greek yogurt obviously is gaining in tremendous popularity. It does have some benefits from the higher protein content, lower sugar content over standard yogurt, but it's still being touted and thought of as a good source of probiotics, so we wanted to test Greek yogurt. A very common and popular Dannon yogurt, which is also another source of probiotics, was tested, and of course MegaSporeBiotic. This test basically put the products, they were counted prior to digestion and then put into a simulated gastric solution, which is a United States Pharmacopoeia standardized gastric solution used across the board in industry. It's a pH of 1.3 for 2 hours that has some bile salts in it as well. And the data shown is % survival of bacterial population before and after digestion. If you look both yogurts, if you look at the data here on the bottom right, both yogurts had virtually no survivability at all. 99.999% of the bacteria were killed in the stomach. If you look at the leading retail product here in the third column, it also had about 99.99% attrition rate in the stomach. None of these products survive the environment, the gastric environment. And if you look at the WHO's definition of what a probiotic is, the very first part of the definition is that it's a live microorganism. It has to be a live microorganism that gets the site of action alive. So none of these products can really be considered to be a probiotic. MegaSporeBiotic, of course, had 100% survival, and that was not surprising to us because these strains are naturally designed to survive the gastrointestinal system. So further new understanding of probiotics, in order for a supplemented probiotic to have an effect, again, it has to have these 3 features. Number one, it has to survive through the GI. If it doesn't survive through the stomach, it's not even a probiotic. So that's the first thing. But it has to be a strain found in the microbiota. There are 1,000 different species found in the human GI out of 35,000 species possible. So if you're looking at a product with 15 different lactobacillus species in there, you have no idea if any of those species belong in your patient's GI or have a position in the patient's GI. And so if they don't, even if they by some miracle make it through the stomach, they won't have a chance of colonizing. The other part of it is it has to be supplemented in concentrations higher than what is found in the microbiota. So in order for a probiotic supplement to cause the necessary stimulation to get the effect, it has to be supplemented in cell counts that are higher than what's found endogenously in the microbiota. If you imagine this, let's say you've got a particular strain of lactobacillus that's being used as a probiotic. That particular lactobacillus strain in the human GI is at a population of 10 billion, 20 billion, or a trillion in many cases. And you supplement 5 billion, 10 billion, you're not really making even a drop in the bucket. Lactobacillus and Bifidobacter species exist in the human GI at as high as 20 to 30 trillion. So if you're putting in a few billion, you're not really making any impression at all on your immune cells and your GI cells. And then the third thing is they have to be able to survive the GI passage and do so naturally. Now, if we are reverse engineering these strains and enteric-coating them and doing directed evolution to try to find versions of the strains that survive the acid more, then they're no longer the wild-type strains that naturally exist in the GI. And so they have to be able to naturally survive the passage, the oral passage through the stomach and get to the GI alive to colonize. So what are the issues for doctors? Look, considering all this, what are the issues for doctors with the current probiotic offerings? Number one, your products are likely mislabeled. They could be mislabeled, but they're more than likely mislabeled with the wrong strains, and in fact, strains that should not even be probiotics could be in there. And there's no way for any doctor to verify this. The potency is questionable. Of course, most of the products tend to have less bacteria than what's claimed on the label, and even whatever's claimed on the label don't even survive the stomach. With no effect, with no positive effect, patients won't continue to take the treatment, and that's loss, of course, in efficacy and treatment of the patient, but also loss of revenue. Probiotics are medicines, and we believe they're very powerful tools for doctors, and it's a tool that doctors can use to really improve many, many different conditions, and Tom will talk more to that effect. But it's really a pharmaceutical, and it should be of pharmaceutical grade with validated and verified strains of validated and verified amounts. There's nothing proprietary for doctors with a lot of the products that you have available to you. Patients can find the same products or very similar versions online for much cheaper, and then you lose the annuity that you've invested in, the time and effort with that patient. And lastly, even if they work, and in some cases some of these standard probiotics do have some effect, but even if they work, the effect is for a very small range of indications. Most very common gastrointestinal disturbance, but as Tom will illustrate, with a true probiotic, one that is highly effective, you'll find that you can use it to help with so many more different indications, things that you'd never even thought about. So what is nature's design for a true supplemental probiotic? Now with our understanding, we know that a lot of what's offered in the market today is really not probiotic. It's really not a probiotic. They don't really have the features of a true probiotic. But we also know probiotics are important and something that humans evolve with. So what is nature's design for a true supplemental probiotic? The most popular, the most well-known are bacterial spores. In particular, bacillus spores. Bacillus spores are the most widely studied and most widely used probiotics outside of the dietary supplement industry. And I say that because these bacillus spores have been commercial products in prescription markets for over 50 years now. Two of the most popular ones, Endrogermina and Bacti subtil, were released in the early 1950s. And these are bacillus spore-based probiotics with very specific dosages that have been used, very widely used for over 50 years. They're also extensively used in agriculture and aquaculture, both to help the growth and the health of aquaculture, or animals, and then in agriculture as well, in terms of birds and cattle and pigs, in reducing mortality rates, increasing weight gain, reducing the amount of antibiotics and hormones that need to be used. So they're used very, very widely in every industry except for the dietary supplement category. And part of that is the availability of verified and branded strains of these products, and that's where we've come in. We've actually licensed these strains, from the university in order to make them available to the dietary supplement community. These strains are all branded and verified. The most widely used and well-studied strains in humans are Bacillus subtilis, Bacillus licheniformis, Bacillus coagulans, Bacillus clausii, and we're introducing a new one that really has not been used very widely in the market, but it's got a very novel feature to it, and that's Bacillus indicus. And that strain is characterized as HU36. Key features of bacterial spores and why they are the ideal naturally designed probiotic. Number one, they form a robust endospore that can really withstand very harsh temperatures, desiccation, low pH, gastric barriers, basically antibiotics, UV radiation, solvents, basically anything. They become inert and they become very robust. They do this as soon as they come out of the GI, and in fact that's a demonstration of the coevolution of these bugs. They actually, their ideal environment is in the GI, but they can use the environment as a vector to transfer from host to host. And so as soon as they're leaving the colon, they go from a vegetative regular cell into this robust spore form so that they can survive in the environment and then be transferred to the next host. They can remain dormant for up to 50 years in the environment. You can dig in the Sahara Desert, 6 feet under the sand, you'll find some of these spores. They colonize very effectively in the human GI. They're naturally designed to colonize. There's actually very specific recognition proteins between the microbial epitopes on the outer cell wall and some of the human GI cells. And so it's a clear demonstration that there's a coevolution. These are not microbes that are newly introduced to GI. They have been around in the human GI for hundreds of thousands of years. They are found as part of normal human commensal flora. Almost everybody will have some level of spores. Unfortunately for us, because we live in a sterile environment and we have a sterilized food system, we really don't get the exposure to these spores that we need to. Our ancestors got tremendous amount of exposure to these spores because they ate off the land. They were hunters and gatherers, and they gathered food, and every root and tuber that they ate contained these spores. All the meat that they ate contained the spores. The animals that they ate contained the spores, because these are universal probiotics that exist in the digestive system of every living animal. It's got a long history of use in industries where the efficacy is closely measured, like pharmaceuticals, and also in aquaculture and agriculture. They wouldn't add these spores into these aquaculture and agriculture industries if they didn't see the very specific health benefits that they got from it. So it's very widely used, verified for over 50 years now. They're extremely safe, and its use as a probiotic is supported by evolution, because they are some of the most unique species of bacteria because they're called biphasic. They have two life cycles. They can exist outside of the human host in the environment, but then they also exist in the human host, which is a very different environment than the outside world. And so they have that biphasic lifestyle where they can exist outside, be stable, use the environment as a vector to transfer it, and the moment they get into the upper GI of the human tract, they understand where they are and they come out of the spore form and turn into a vegetative thriving cell. So they are clearly what nature has designed to be what your true probiotic is. Remember that after birth, we get all the lactobacillus and bifidobacter species we're ever going to get. We get it from mom. We get it through the amniotic fluid and through the cord blood itself. We get it through passage through the vaginal canal during birth, and then we get it through mother's milk. After that, those species are not found anywhere in the environment, so humans are not designed to consume those species on a regular basis. Spores, however, are found as normal environmental organisms all over the place, and some subclasses of spores have developed a biphasic lifestyle where they exist in the environment and also thrive in the GI. So these are very specialized, true commensal organisms designed by nature to be probiotics. Here's a quick illustration of what the spore looks like. It's got a thick outer spore coat and an inner spore where the genetic material is. This cell basically becomes dehydrated, and the outer spore coat is a very thick coating that protects the cell. It has little sensors sticking out to look for what environment it's in. As soon as it detects the environment it's in, the spore coat comes off and the cell becomes a vegetative cell. So the question is, are there any effective spore-based probiotics on the market today? Understanding that spores really are true probiotics, are there any spore-based probiotics in the market today? And if you search around, you'll find that there really isn't until now, until we introduce MegaSporeBiotic, which is the first all-spore carotenoid-rich probiotic consortium formulation. And again, this is sold through doctors only, and Dr. Tom Bain will take it from here.
Okay. You guys on that going again like you did last time. Hopefully we've got that corrected on our end. What I plan to do with the rest of our time here is go through some of the specifics of MegaSpore and some of the clinical indications of it, and then answer some of your questions. So what we're looking at first is MegaSporeBiotic. It's a complete probiotic. So we developed this product under the advisement of spore expert Dr. Simon Cutting. It was designed by a practitioner for practitioners. We're utilizing verified, branded, and registered strains, which is the only way to ensure that the strains that you're using are, in fact, characterized correctly. This is the only dietary supplement that contains the probiotic Bacillus clausi. And this is a strain that's been tested for over 50 years. It's the largest selling prescription probiotic in the world. We've got 5 spores existing in a consortium like they do in nature. In nature, these spores don't exist as singular strains. They exist as a group where they work in synergy with each other. All our strains are produced in a certified GMP facility under drug manufacturing guidelines, and all the strains are prescription grade and licensed from Royal Holloway, London University. We're delivering 4 billion live probiotic cells daily, which matches or exceeds all prescription products of these same strains. We're blended with a prebiotic to enhance colonization. That prebiotic is a cellulose from organic acacia root. We're guaranteeing 100% spore format for maximum survivability and precise dosing. And on top of everything else, we're delivering the first carotenoid-rich probiotic, a probiotic that's actually going to be making these precious antioxidants right at the site of absorption. So we've got numerous studies that show a long history of colonization of the spores in our digestive tract. In fact, it's the ability of the spore to resporulate as it moves through our digestive tract that gives us the stimulatory benefit to our gut. It actually is responsible for the development of our gut immune system. The bacillus spores are found in the GI of insects, animals, humans. And what's interesting is that these bugs are typically referred to in the literature as soil-based organisms, when in fact they do use the soil as a vector to move from host to host, but that's when they're in their inert state. And so where they're their most active state is actually in the digestive tract. So the research shows that these spores produce significant numbers of antibiotics. So in other words, they're able to go into the intestinal tract, police the intestines. And if there are pathogenic organisms or pathogenic bacteria in the digestive tract, they're able to actually manufacture antibiotics to rid the digestive tract of those unwanted infections. They also use competitive exclusion with pathogenic organisms like yeast and bacteria. And so what that means is that they will fight for space on the digestive tract with pathogenic organisms like Candida, like C. diff, and some of the other things that Karan had alluded to earlier. So very potent immune modulators. So immune modulation is bringing your immune system up when it's down or down when it's up. And so the only way that we know of to modulate the immune system is to actually stimulate the gut immune system. And we're able to do that with MegaSpore with each dose. And so that's what I alluded to there. So the MegaSpore also is an incredible digestive aid. These spore bacteria produce the full gamut of digestive enzymes to help your body digest foods. And they're very good for correcting a dysbiosis. So up to this point, we've heard what an incredible probiotic this MegaSpore product is. But when you add this next piece, it really pushes this product beyond all the competitors. And that is that these bugs act like tiny little nutrient factories inside your digestive tract. So once they get to the other side of your stomach and into the small intestine and into the large intestine, they actually will produce the full spectrum of B vitamins. They'll produce nattokinase, vitamin K2. And what the Bacillus indicus has been patented for is the biological delivery of carotenoids. And so this is a complete game changer in the field of probiotics in that not only are we competitively excluding pathogenic organisms, but we're actually producing nutrients that are difficult for the body to absorb in the first place right at the site of absorption. So some of the other things that the Bacillus probiotics do is they help with detoxification, they help with cholesterol reduction, and some crosstalk with human cells where the spores actually communicate with our cells for them to perform functions such as seen in the heat shock peptide. So why these 5 strains? So these are the 5 most studied spore probiotics available. And so we'll go through each one of them individually. The Bacillus subtilis is a very potent immune stimulator. It's widely used, safe, highly effective, produces over 12 antibiotics, It produces nattokinase and K2, and it by itself is very important for both the development of the gut immune system and the daily stimulation of the gut immune system. Bacillus indicus is what we've spent a little bit of time talking about, produces high levels of carotenoids, alpha carotene, beta carotene, astaxanthin, zeaxanthin, lycopene, and lutein are the carotenoids that have been identified to be produced by this very interesting bacillus spore. These are considered the most effective antioxidants on the market because they don't have to go through the survivability issues of getting through the stomach acid like other antioxidants. Bacillus clausii is the most widely used probiotic drug in the world. It's a very potent stimulator. It's antibiotic resistant. So we actually recommend that you use MegaSporeBiotic when you're taking or when a patient's taking antibiotics because it's very effective at stopping antibiotic-induced diarrhea and secondary infections like C. diff from overuse of antibiotics. Bacillus licheniformis produces the antibiotic bacitracin. It also produces protease and other enzymes to help with digestion. And it produces the full spectrum of B vitamins. Bacillus coagulans, very well studied, long history of use, very effective against IBS and Crohn's disease because of its ability to produce the L-positive optical form of lactic acid. It's also a potent immune stimulator. So, keys to efficacy. So here's the key thing. So these organisms exist in your digestive tract somewhere in the range of 2 to 3 million. So when we're supplementing 4 billion of these organisms, we're giving you 2,000 times what naturally exists. As Karan alluded to earlier, when you're looking at lactobacillus and bifidobacteria-based products, you're looking at 20 to 30 billion of these 20 to 30 trillion of these organisms existing in your digestive tract. So even if you could get 50 billion or 100 billion past the stomach acid, it's not enough to create that stimulatory benefit to your gut immune system. We provide these organisms in a consortium as they exist in nature, and we can guarantee that they're all spore pharmaceutical grade strains. As cool as these bugs are, if they're not in spore form, they won't survive the stomach acid either. And so it's critical that we can ensure that they're 100% spore form. And that's another place where you'll see our product differing from other spore-based products in the marketplace, is that most of those products are upwards of 30% spore form at best. So there's upwards of 70% at least. So I want to kind of go through this a little quicker. We're short on time here. So you guys can look through these slides on your own. A lot of these slides that I'm going through now just kind of go into the specific features of the antioxidant qualities of MegaSporeBiotic. I don't mean to make light of that, but I do want to be able to get into some of the clinical stuff. So just to review, we're at the required features of a probiotic. Rule number 1, as Karan went through, must have a position in the microbiota. MegaSpore qualifies. All strains of MegaSpore are humorized and have a firm place in the microbiota. Must have sufficient numbers to colonize and stimulatory benefit. As I said, we're dosed at a concentration 2,000 times higher than the native population of the GI tract. And supplemented probiotics by evolution should naturally occur in the environment as well as the gastrointestinal tract. MegaSpore is based on nature's very own probiotic. It exists in the environment as well as in the GI tract. We are capturing the power of millions of years of evolution. So, indications. So, immune modulation is one of the qualities that MegaSpore provides, which would be great for patients with allergies, asthma, rheumatoid arthritis, or different types of immune dysfunctions. So, MegaSpore can stimulate the peripheral T lymphocytes and B lymphocytes, which is critical to help fight colds, flus, sinus infections, ear infections, gut infections. It's very good for improving urogenital health and flora, for treatment of urinary tract infections, able to control gut microbial growth, which is great for post-antibiotic therapy, also great for small intestinal bacterial overgrowth. Reduce gut inflammation, which is beneficial for Crohn's, IBS, ulcerative colitis, and even obesity. detoxification of the intestinal tract that would be necessary in patients with skin afflictions and fatigue, improved digestion, improved nutrient absorption for patients with fatigue, malaise, athletes, osteoporosis, cardiovascular disease. It's very good for the reduction of cholesterol. And we're also looking at, with the production of these critical nutrients, we're looking at a reduction in oxidative stress and overall health improvement. So, we're now at the end of our discussion. We want to answer some of your questions. So Beth Ellen typed in, sorry for confusion, but you're saying you're doing 4 billion, and that's 2,000 times the number present in the GI tract, but then say that their presence in the GI tract is in the trillions. Okay, so to be specific, Lactobacillus and bifidobacteria species exist in the GI tract somewhere in the 10 to 30 trillion with a T. Those organisms are naturally occurring organisms that we get from our mother and from the first year of development. These spore-based bacteria that exist in our environment exist in our gut in the 2 to 3 million. They're transient organisms, and they move through our digestive tract performing certain duties, and they're there in a number somewhere of 2 to 3 million. When we supplement 4 billion, that's 2,000 times what naturally exists there. In order to do 2,000 times what naturally exists of Lactobacillus and bifidobacteria, you'd have to multiply 20 trillion times 2,000. And so no product is able to provide those. And besides, we've already shown that they're not able to survive the GI tract. So, any other questions? Okay, so Sonya's asking, what are some of the reported changes I've seen with patients? So to date, I've got about 800 patients that are taking the product. And what we're seeing is really, really good results with primary digestive issues. Issues like IBS, bloating gas, ulcerative colitis, Crohn's disease. We're seeing pretty profound effects in a short period of time. I think this is a good place to allude to the dosing. 85% of the patients that you're going to give the product to are going to do fantastic with the product. The 15% that have detox symptoms after taking the product, those symptoms are mitigated by titrating them up slowly. So the clinical dose is 2 capsules a day. With patients where you are suspicious of a chronic yeast type presentation, it would be in your best interest to titrate them up slowly. Start them at 1 capsule every 2 or 3 days, and then get them to a capsule every other day, and then a capsule every day, and then even up into the 2 capsules a day. And so that's how we are being able to see who the people that are reacting, being able to see them sooner. Patients that are having detox reactions typically are experiencing some cramping, a little bit of loose stools. And those symptoms go away immediately when they stop taking the product. And then if we slowly dose them back up, they're able to tolerate the product pretty well. Okay, so let me get Karan in here. I've muted him here.
I'm here. Can you hear me?
Yeah. Karan, you want to answer some of those questions?
Yeah, well, Beth asked if these products are available to nutritionists or just physicians. We're making them available to every healthcare practitioner, so nutritionists. We're working with chiropractors, DOs, MDs, naturopathic physicians. So we're making it available to a broad range of healthcare practitioners. So absolutely to nutritionists, and in fact, nutritionists, could be one of the most important tools. A clinical nutritionist that we work with very well has some of the Blackhawks players in Chicago on the product, and they seem to be doing well with it. So that's an interesting little tidbit. Kenneth, you asked if there's specific research showing these indications in the real world as being effective. You say cholesterol reduction is not a real issue. Well, yeah, so there are specific research papers that show the effect, some of these clinical indications for some of the strains. And so the idea here is that um, in the pharmaceutical industry, one or two of these strains have been used together. In the agriculture, aquaculture, in some cases, two or three. What we've done is we've taken the most well-studied strains and looked at the clinical doses of each of those strains in use over the last 50 years and put together a consortium of all five of these strains. And each one of these strains are in a clinical dose in the product. They're not just in there so they could show up on the label. Each one of these indications has some sort of studies to demonstrate that. Like, for example, immune stimulation of peripheral T lymphocytes and B lymphocytes. At least two of the strains have shown very key immune proliferation of T and B lymphocytes, which reduces the duration of colds and flus. SIBO with small intestinal bowel overgrowth, Bacillus clausii, has got a very powerful published study on looking at small intestinal bowel overgrowth. And some of these, so we actually have probably 25 published papers that we can send you as a research packet that cover pretty much all of these indications. Now, many of these are, structure function type indications, like immune modulation. There isn't a very specific study on rheumatoid arthritis and these spores, but we know that RA is a autoimmune condition with certain immune dysfunctions, and then there are studies that show that these bacteria support these or help alleviate these immune dysfunctions. So, you can show improvement in patients with RA with these particular types of indications. So we can send you a collection of research papers that basically takes care of most of these. Let me look at Tom. Did you want to take the next questions?
Yeah, I'll take these next questions. So Sonia is saying that after dosing the MegaSpore, so she's been taking the product obviously, that she's having some more gas and nausea and some pain at night. So it's typical clinical presentation we see with people with yeast overgrowth, is that there's more bloating and more gas and some slight nausea. And so what I would recommend in that case is that you decrease the dose. Sometimes that's decreasing the quantity that you take daily, or sometimes that's keeping the dose that you're taking the same, but taking the product, say, every other day, or something like that. So you should have a dose that works for you. If you start at a quarter of a capsule every other day, and that's where you're asymptomatic, then that's your starting point. And you stay with that dose for a period of time, and then you slowly start to move it up. And so I find more frequently it's not the amount that the individual's taking at that time. If they're taking a capsule a day, that's not too much, but that every day is too much, and that they would be better off leaving the dose at one capsule but taking it every other day or every third day. But you have to experiment. Taking a fourth of a capsule or a half a capsule is done by just opening up the capsule and pouring a little bit of the powder into some food or into some water and drinking it, or pouring it straight onto your tongue. Liz had a question. Thanks for coming, Liz. What about long-term maintenance dosing? So again, that kind of goes to what the situation is for the individual. If you look at the information we've given you today, you see how much the first year, the development, and then the first year of life, how critical and how important that is. And we can't necessarily give you that back. So if that is the reason why your gut immune system is underdeveloped and your body's expressing itself with digestive symptoms, then your long-term dosing might be different than somebody who is, for example, taking it for the antioxidant benefits. Personally, I take it daily because I'm looking for the benefits of the antioxidants. Those are cancer preventative. Those are eye, prostate, breast health. Those are antioxidants that are very difficult to get from the diet. So I use a daily dose for that perspective. When it comes to looking at someone where you're dosing them because of chronic digestive issues, it's possible that they would need it, daily. It's also possible that they would need it weekly. So instead of taking it daily, every day, 1 capsule or 2 capsules, that they need 2 capsules 4 or 5 days a week. It's something that you really need to experiment with as a patient and see. If you're someone that uses muscle testing or something like that, that's a great way to monitor yourself on a daily basis. But if not, you should be able to use a titration up to the clinical doses that we talked about. I think of maintenance as, it's changed the audio one way. I think of maintenance as a continual thing. I'm going to keep talking just in case there is audio, but I'm hearing that it's gone. So continual dosing for depends on what types of symptoms that you would be seeing. There it is. Okay. All right, so I'm still here. All right, so one more question from Kenneth. Is this product the answer when some take probiotics and feel the difference and others take the same probiotic and has no change? Well, one of the problems with standard probiotic products is we don't dispute that some of the standard lactobacillus and bifidobacteria-based products create some type of symptom reduction. We're not disputing that. What we're disputing is that the reason is because these bacteria go and live in the digestive tract. And so with certain probiotics where people feel a difference, typically it's the ferment that's creating the change and not necessarily the bacteria. There's also been some studies that show that the dead bacteria can infrequently but still at times create some immune system stimulation in the gut. And so some of these other probiotic products where you have patients who feel benefits, or somebody says, I feel great after I eat yogurt, it's typically more the ferment, the waste products that the bugs are making while they're in development, and not necessarily that these bugs are going and living in the digestive tract. So let me bring Karan back here. I think we're about finished. Karan, anything else you want to add?
Yeah, as far as the daily use of these types of probiotics, we have to look at what nature has done. So basically, our ancestors over the last 100,000 years have been consuming these particular types of strains with every meal. So it becomes part of your basic nutrition. We can give people clinical doses for acute issues or clinical doses for chronic issues, but they should be some part of your daily nutrition, if not every other day, certainly at some point in your dietary practices, because these bugs are designed to be transient organisms where they don't stay forever in the GI, which is really a feature of a true probiotic. They should come in there, perform a function, and leave, and then wait to be reintroduced again. And so really, we think of this as not just a probiotic in a general sense, but it's part of your essential nutrients, no different than taking vitamins, protein, and fish oil and things like that. It should be part of a daily nutrition regimen for even healthy people, because there's enough studies that show that these spores play critical roles in immune development and immune function and cellular function in the GI, that they're required by our bodies, and we've basically eliminated them from our food source because we've got a sterilized food source. So it's important for them to be part of your nutritional system.